Welcome to everyone, and thanks for attending this webinar. I am Paola Lazzeri, and I'm Field Application Specialist for Sterility Assurance and Microbiology at Particle Measuring Systems. This presentation is focused on the indications that have been included in the latest revision of Annex 1 with specific regard to single-use systems. Other similar presentations are available to deeply evaluate step by step different aspects of the new Annex 1 draft with particular regard to Revision 12. This webinar represents the opportunity to better understand the current state of the regulatory position. Therefore, this content cannot be used to defend any regulatory position. The new Annex 1 introduces the concept of single-use system as possible alternatives to reusable equipment. The advantages of single-use systems were compared to reusable equipment with regards to sterility assurance are often obvious. Therefore, when a new equipment is implemented within pharma areas, any possible risk should be avoided. And so, the new Annex 1 describes in details any specific risk associated to single-use systems in order to support pharma users in the evaluation of new disposable items. So, now, let's evaluate point by point the indication reported in Annex 1 and do the exercise together to evaluate the complete compliance of BioCapt Single Use with the new regulatory approach. The first aspect underlined by the new Annex 1 to evaluate a single-use system concerns the component of the item, to exclude any risk related to absorption, leachable, and extractable. Speaking about this point, no additional risk has to be evaluated referring to BioCapt Single Use, because the plastic component of the device is made with the same plastic component of standard agar plates, that polystyrene. So, we can confirm that BioCapt Single Use is in compliance with this key point, and we can proceed with our evaluation. Any risk caused by the fragility of a single-use system should be avoided. Speaking about BioCapt Single Use, of course, if we compare the stainless steel with plastic, we could have to consider the risk of fragility. However, the fragility risk must be managed considering the whole system. Thinking about the risks related to handling procedures of agar plates in combination with the stainless steel impactor, we can surely confirm that BioCapt Single Use cannot be considered more fragile than the traditional solution of stainless steel impactor plus agar plates. Why? Because of its unique feature. It is a complete and closed system. So, even if we consider the possible fall of the sampling device in a filling line, using the BioCapt Single Use, we would have no risk of contamination caused by the agar. We cannot help thinking about the consequences caused by the fall of an agar plate. So, green flag for us, and go on. The considerations shared before are confirmed by reading the third indication concerning single-use system. In fact, the Annex clearly requests to consider the number of manual operations requested for using a single-use system and their complexity in comparison with the existing solution. No problem regarding BioCapt Single Use. Only advantages. BioCapt Single Use only needs a plug-in, plug-out procedures. So, one safe step instead of several manual operations. So, our third green flag is positioned. Speaking about the next point, we speak about the design of assembly. Well, the design of the slits is exactly the same for BioCapt stainless steel and for BioCapt single use. The patented design of BioCapt is certified for its physical and biological effectiveness, and the dimension of the slits of BioCapt single use are verified on 100% of the items. Then, I'd like to underline that a unique advantage of BioCapt single use concerns the exact positioning of the agar. In fact, BioCapt single use guarantees the respect of the distance between the agar surface and the top of the impactor. As we all know, this distance represents a critical parameter concerning the microbial efficiency. So, another okay, we can go on. The fifth point mentioned by the Annex 1 is specific for filter, so it is not applicable to BioCapt single use. Now, we have some recommendation concerning integrity of single-use system and the evaluation of any leakage risk. About these aspects, we simply have to point out that BioCapt single use contains agar, and that, consequently, any eventual integrity failure shall be detected as an extraneous element in the agar. We can say that every manufacturing batch of BioCapt single use is a sort of media field for us. So, next point considered by the Annex 1 is the potential for compromising the system at the point of opening the outer package. Also regarding this aspect, BioCapt single use shows to be in fully compliance with the regulatory request because it is protected by 4 bags. This packaging allows operator to take the package through each level of classified areas by simply removing 1 bag at a time. 2 large bags facilitate the introduction of BioCapt single use from not classified into aseptic areas and protects the double-bagged disposable. So, finally, any document concerning the quality of BioCapt single use, including the assessing of PMS as a supplier and the studies concerning particle release caused by the opening of the protective bags of BioCapt single use, are collected and available for you in the BioCapt single use validation manual. So, at the end of our evaluation regarding the recommendation concerning single-use system by the Annex 1, we can confirm that BioCapt single use meets and exceeds the auditor's expectations. Let me say thanks to all of you for attending this webinar. You can find other webinars and papers in Particle Measuring System website, in particular in the Particle Measuring System Knowledge Center. Thanks again. Looking forward to the pleasure of supporting you for any future request.